Brian, tell me first of all about yourself at Cleveland Clinic, your lab, what you're doing and everything.
So I'm Brian Rini, I'm a medical oncologist from Cleveland Clinic, and I do clinical and translational work in kidney cancer.
So how did you get on to looking at this vaccine? Because you're looking at a multi-peptide vaccine in kidney cancer. What are you doing?
So this is Immatics is the name of the company, and they developed over many years this methodology for deriving peptides from tumor cells, peptides that are specific for tumor cells and not normal cells, and further testing their immunogenicity. So they basically came up with a cocktail of peptides that, when given to patients, are felt to generate a relevant anti-tumor immune response.
Now, metastatic renal cell cancer is a difficult one to tackle, isn't it, with the vaccine?
It is. I mean, it's a tumor that's considered immunogenic, kind of like melanoma. It's always had immunotherapy in the background. Interleukin-2 was the first drug approved for that disease 25 years ago. Then came the wave of targeted therapies like sunitinib, and now we're sort of turning back to immunotherapy.
Now, you've been combining this therapy with sunitinib. What happened?
So the trial looked at previously untreated metastatic kidney cancer patients, so this was their first therapy. They either got sunitinib alone, which is a standard of care, it's a VEGF receptor inhibitor, or they got sunitinib plus this vaccine. And the concept was that sunitinib would provide disease control, the vaccine would provide an immune response. There's also data to support that sunitinib has favorable immunomodulatory activity, so that it might actually make the vaccine work better.
So there was some hope that sunitinib might have helped the immune processes in a similar way to perhaps the checkpoint inhibition process do, or a different way, but...
A different way, but a similar result,
idea.
A similar idea, yeah, yes. Similar concept. There are clinical and preclinical data that certain cell populations, certain immunosuppressive cell populations are reduced when patients are treated with sunitinib.
And what happened?
And unfortunately, the trial did not show an overall survival benefit. There's really no difference in survival among the arms. Both arms did extremely well. Progression-free survival was 15 months, which is very long, and overall survival was very long as well. So all the patients did well, and the vaccine did not provide an additional advantage.
Where do you think this leaves multi-peptide vaccines in the context of renal cell carcinoma?
I think vaccines in all of oncology have struggled, I think, not because they can't generate an immune response, but the quantity of that response has always been inadequate, inadequate enough to have clinical relevance. And I think this trial, redemonstrates that. But now that there are checkpoint inhibitors, there are other inhibitors of molecules like OX40, et cetera, again, there's sort of renewed interest in immunotherapy, and I think some of those molecules, some of those drugs in combination may sort of revive vaccines to provide some specificity to the immune response.
So what should doctors take home from this now?
It's not a new treatment option because there was no advantage. Again, sunitinib remains a standard of care. There's a lot of development of immunotherapy in kidney cancer going on right now, and so I think vaccines will continue to be explored. There's other trials out there, including other large trials, but we're really sort of waiting, again, for those newer immunotherapies to make an impact.